Hi, this is Remembering the Past, the show where we talk about people who've died recently who've had a profound effect on our history, our society, or our culture. And in our ongoing series on Nobel Prize winners, tonight we're going to talk about Dr. Thomas Stites, who died recently at the age of 78. Dr. Stites was awarded one-third of the Nobel Prize in Chemistry in 2009 for his work using X-ray crystallography to figure out the structure of ribosomes, the intracellular structures that translate genetic code into protein. The other two award winners that year were Venkatraman Ramakrishnan and Yonath, who also did work in ribosome research. According to NobelPrize.org, an organism's vital functions are managed by large complex protein molecules produced in cells' ribosomes. Their genetic information from messenger RNA is translated into chains of amino acids that then build proteins. Using a method known as X-ray crystallography, Thomas Stites and other researchers were able to collaborate to map the structure of ribosomes made up of hundreds of thousands of atoms in 2000. Among other applications, this has been important in the production of antibiotics, and I might also add, in determining the mechanisms of antibiotic resistance. So in layman's terms, the ribosome is that part of the cell which takes the genetic coding for building proteins, turns it into a blueprint, and that blueprint becomes the pattern for amino acids in a specific sequence, and the ribosome then assembles those amino acids into that sequence, and they become the proteins that are vital for every function in the cell. Obviously, in bacteria, if you can hinder that mechanism in the bacteria ribosomes, you can destroy the bacteria, which is what some antibiotics do. And being able to actually visualize that was the key to Dr. Stites' work. Here's Dr. Stites receiving his 2009 Nobel Prize Award along with his co-laureates in Stockholm.
Professors Ramakrishnan, Stites and Jonath, you are being awarded the Nobel Prize in Chemistry for the crystallographic determination of ribosome structures at high resolution, and for the skillful use of these structures to clarify fundamental and medically important aspects of ribosome function. On behalf of the Royal Swedish Academy of Sciences, I wish to convey to you our warmest congratulations, and I now ask you to step forward to receive your Nobel Prizes from the hands of His Majesty the King.
Well, Thomas Stites was a nice Midwestern boy born in Milwaukee. His family moved to Wauwatosa. He was a proud 1958 graduate of Wauwatosa High, and then he got a scholarship to Lawrence University in Appleton in northeastern Wisconsin. From there, he moved to Harvard, and it was at Harvard he heard a lecture by Max Perutz, who was awarded the 1962 Nobel Prize in Chemistry along with John Kendrew. And that lecture changed Dr. Stites' life.
My passion, and really my passion for trying to understand how macromolecules function in biology and understanding biology at the molecular level, started from a lecture that I heard by Max Perutz in 1963. It was a Dunham lecture at Harvard, and he talked about his structural studies on hemoglobin and Kendrew's studies on myoglobin. Now, they had won the Nobel Prize in Chemistry just the year before for their determining these structures. And so Max showed a stereo slide of myoglobin in the auditorium with at least as many people as here, or maybe even more. And it was a very large screen over his head. And we all had colored glasses on, and he went back and forth. And you know how that works. And then all of a sudden, it popped into 3 dimensions. And the audience went, whoa. I don't expect that'll happen here. But none of us had ever seen a molecule, a protein, in 3 dimensions. And that was when I knew that's what I wanted to do. I wanted to understand biological molecules at the atomic level in 3 dimensions, and that's what I've been pursuing.
Here in a phone interview with the Nobel officials from Stockholm, Dr. Stites talks about what it was like to see the 3-dimensional structure of a small organelle inside the cell.
What I found the most exciting thing about the ribosome was seeing a structure, seeing a large macromolecular assembly that I had no idea what it would look like. I have to say it was the most exciting time in science that I've had, like getting to the top of Mount Everest and seeing the view. It started coming out the earlier stages of it in '98, and then of course there's always communication back and forth about how to do things, and it all happened at once after many years of not happening. So it was great. So.
Do you feel that we now have a good picture of the way that what ribosome works?
Oh, I think it's extraordinarily good picture. I mean, there's always more that one can do, of course.
Well, there have been significant practical implications to Dr. Steitz's discovery. Antibiotics work against bacteria in different ways. Some work against the cell wall, some work against the nucleus, and some work against ribosomes. When Dr. Steitz was able to visualize the three-dimensional structure of the ribosome, all it took was to add different antibiotics to bacterial media and study the ribosomes of the bacteria. They would see which antibiotics bound to the ribosomes or interfered with the protein synthesis that the ribosomes were responsible for, and in that way they could determine which antibiotics retarded bacterial growth or actually killed bacteria by working against ribosomes. Not every antibiotic did that, but certain ones did. Along with this, they could study the ribosome-antibiotic interaction within the bacteria and study how bacteria would form their own defenses against the antibiotic. So the bacteria might try and modify the antibiotic with a different chemical group, or they might try and break up the antibiotic, and you could see that interaction at the ribosomal level. Here Dr. Steitz talks about the importance of ribosome structure in discovering mechanisms of bacterial resistance to antibiotics.
It's led to insights in a variety of things, and the most, I think, the one that will have the most profound effect on the public at large is the ability to use this structural information to design new antibiotics that are effective against disease-resistant strains. It's a rich environment for designing antibiotics, and it's the major target of antibiotics in general anyhow. So, and the structure has been very illuminating. Now, that wasn't our original goal in solving the structure. I wanted to understand how it worked.
Yes.
But this was sort of a little side benefit of learning the structure. This is why I think basic research is very important, because it can have applied consequences that you didn't even think about. And there was an article in the New York Times about a year ago, 2 years ago, that said there were 20,000 people a year who died of MRSA infections in U.S. hospitals alone. That's a lot of people.
As with many of the Nobel laureates, Dr. Steitz was a modest man, and he talked about the importance of basic research to the extension of practical research and how it was vital to begin with basic research.
It's a large group that works together, but the thing that I appreciate is the recognition for the group. And I take the prize to be the recognition that the work that the group did on the ribosome structure is appreciated, and it gives us an opportunity to convey how important this basic research, which is the real reason why we did it, is going to have applications. In fact, it's being used for drug design, and it's going to be very important. I never started working on the ribosome because I thought of its practical consequence. In fact, I should have known that the ribosome was a major target of antibiotics, but I didn't really. It was only after we started that I realized this, and we started working on it. And I must say, one of my concerns about what I see with funding agencies increasingly is this desire to have translational research. Translational research as applied to the ribosome is fine, but I think translational research in the sense that you want to target your research on doing something practical, I think it's the wrong way around. I think you want to understand the world and then use that information to solve the problems that it enables.
There was actually some mild controversy about the 2009 Nobel Prize in Chemistry. There was about too much biology and not enough about chemistry. And here Dr. Stites addresses that controversy.
Well, of course, all biology is chemistry, right? I mean, you can't understand biology without understanding chemistry. I mean, that's the answer, right? And, I wanted to know how peptide bonds are made. That's chemistry. A little orbital is attacking something else. What's facilitated? Oh, by the way, RNA is not what you would choose for a catalyst. If you've been trained in some organic chemistry course and you look at the pKa's, that's chemistry, of the bases, you say, that's going to be lousy. How can this happen? So the issue of how an RNA can catalyze a chemical reaction, if that isn't chemistry, well, I guess I don't understand what chemistry is.
Another interesting fact about Dr. Stites is when he was at Harvard, he met Joan while they were both graduate students in biochemistry and molecular biology. When they left Harvard, they moved together to Yale, where Dr. Stites did the work that he was awarded the Nobel Prize for, and interestingly enough, his wife has become one of the top molecular biologists in the world, and she was awarded this year's Lasker Prize, which is one of the top prizes awarded in medical science. It's conceivable that at some point in the future she may also be awarded a Nobel Prize. Well, one of the things I want to do, if possible, with all our Nobel Prize winners is get their advice for young students. Dr. Stites was a musician when he was at Wauwatosa High, and he thought about going into music when he was at Lawrence University, only to be talked out of it by one of his professors. Here he gives his view on what young people should pursue.
I think the advice I would give is to follow what you actually want to do, and don't pay attention to what everybody else might say, or contemporaries. Perhaps they have the right idea, but follow your passion and do those things that you would think would be of greatest interest to you. Don't follow the money, follow your interest. That's what I did, and I'm very happy. I had this decision, should I go to music or should I go to science? Either way would have been fun, but following science was very.
Well, as I said, Dr. Stites went to Lawrence University, which is a beautiful little university in Appleton, Wisconsin, not too far from Green Bay. It's primarily a liberal arts university, but recently they named the Hall of Science after him, and he attended the naming ceremony where he said this.
Yeah, you never know where you're going to wind up, no matter what you're doing or where you are. You just follow what seems like an interesting direction and see what happens.
Well, I'm going to close on that note. I want to thank my producer and IT genius, Sid Tepps. And in closing tonight, I want to note that Dr. Stites spent 3 years at the Medical Research Council Laboratory of Molecular Biology in Cambridge with a number of Nobel Prize winners, including Max Perutz, John Kendrew, those are the guys who inspired him at Harvard, Francis Crick of Watson and Crick DNA fame, Sidney Brenner, Frederick Sanger, and Richard Henderson. These are all among the great names in medicine and chemistry in the 20th century. Dr. Stites wrote, perhaps the most remarkable and unique feature of the laboratory was the canteen located on the top floor, which provided coffee in the morning, lunch after midday, and tea in the afternoon. The attraction was definitely not the bangers or the toad in the hole or other culinary opportunities, but sitting down with a random collection of lab directors, postdocs, and graduate students and talking about science. Initially, I wondered how anyone got any experiments done since they were spending so much time in the canteen, and then I realized that the many discussions reduced the number of unwise or unnecessary experiments that were done and enhanced the good ones. Well, my reading of that is that it's sort of a happy talk. And as a final tribute to Dr. Stites, we're going to take that song from the 1949 musical South Pacific, done by the jazz great Nancy Wilson. And that's Cannonball Adderley on the cornet.
Happy talk, keep talking happy talk. Talk about things you'd like to do. You gotta have a dream. If you don't have a dream, how you gonna have a dream come true?